Welcome to the Virulent Vortex, a podcast in which we discuss infectious disease across scales, from pathogens and molecules to populations, pandemics, and everything in between. My name is Jaap de Rode. I'm a biology professor here at Emory University. And with me today is Ashley Alexander, who is a graduate student in the Population Biology, Ecology, and Evolution program here at Emory, and about to defend her thesis tomorrow.
Yeah.
Ashley, welcome to the show.
Thank you. And thank you for the great timing.
Yeah, it's great. So you're going to be done tomorrow with your thesis.
Yeah.
And so let's talk about it. But what were you researching and what did you find?
Yeah. So my research had two different parts to it. I studied both the intraspecific interactions of Staph aureus and cystic fibrosis infections. And I also studied interspecific interactions between Staph aureus and Pseudomonas aeruginosa, which are the two most common pathogens in cystic fibrosis.
Okay. So let's talk about intra and interspecific. Yeah. So these are basically ecological terms where we mean it's competition between members of the same species or competition between members of different species generally.
Yeah, exactly.
Right? So you're applying this to bacteria in humans, in the lung?
Yeah. So tomorrow I'll talk more about how I've approached this question of how species interact within and between other species in infections from sort of an ecological standpoint and thinking about how microbial communities are ecosystems, just like forests and savannas. And those interactions within and between species really dictate how that community forms and changes over time and can tell us a little bit more about how we might better treat and influence that development of the community in ways that are more beneficial to the patient and maybe easier to treat down the line too.
Right, so that sounds really intriguing. So ecological principles that people have tried to understand, say on the African savanna where you have zebras and antelope competing for plants and then there's predators such as lions and hyenas that try to eat them. So we'll get back to that. But before we do that, just a question about cystic fibrosis. Cystic fibrosis itself is a genetic disease, right?
Correct.
It's not an infectious disease.
Correct, yeah. So cystic fibrosis is a genetic condition that individuals who have it are born with. I think the estimate is that about 25% of people carry a non-functional version of the CFTR gene. And if you happen to inherit two non-functional copies, then you have the condition of cystic fibrosis. Right. And so there are many ways that disease plays out in individuals. And I focus on the infectious disease component where people who have CF are more likely to acquire respiratory infections that are lifelong sometimes and are the major cause of morbidity and mortality for people with CF.
All right, so they're more likely to get these bacterial infections. Is that because they cannot clear it through the mucus production and removal from the lungs?
Yeah, essentially. So they have less active cilia and so less active clearing of fluid, and that leads to a buildup of sputum in their lungs, which enables bacteria to grow in the environment.
Right. Okay, and so then you're talking specifically about two different bacteria, both of which are involved in infectious diseases.
Yes.
But then you're looking at their interaction as well. So introduce both bacteria and tell us a little bit about them, and then tell us, what they do to each other.
Yeah, definitely. So you've probably heard of staph or Staph aureus, or MRSA also is something we encounter a lot in healthcare settings outside of CF. So Staph aureus is a highly human-associated pathogen. It's been evolving alongside humans for probably since humans were humans and before that. It has very many human-specific adaptations and toxins that it produces that only affect humans. And it's a pretty impressive pathogen. It actually can infect all of our tissue types, from our nasal passageways to our bones. And we estimate that about 30 to 50% of people carry staph with them commensally, so it's not causing disease. Staph is a really common pathogen, especially when patients are young. And then Pseudomonas aeruginosa is also a really cool pathogen. It's kind of
cool.
It's very cool.
So this is you as a scientist, not as someone who is concerned with the well-being of humans.
Correct. if a microbe could be charismatic, I would say Pseudomonas certainly is. It appears blue on our plates, and it has kind of a grape-like aroma. And so it's one of those pathogens that most people can clear quite quickly if they are, healthy. But if it gets into places like eyes, it can cause really serious infections. And in that instance of contaminated eye drops, there's been quite a few instances of blindness and even death from that. So it's a pretty serious pathogen in its own right, but it's most well known as being a really serious pathogen for people with CF. And in particular, causing, severe buildup of biofilms and being really difficult to eradicate because it's naturally drug resistant.
Can you tell us what a biofilm is?
Yeah, so a biofilm is a bacteria-produced structure that it's basically like a slime. So you've probably encountered it in things like your bathroom and in pipes. It's just a buildup of secreted proteins and sugars, and even things like DNA can build up that structure of a biofilm.
Just a bacterial slimy layer.
Yeah.
Okay, and so Pseudomonas does this in the lungs. And so then what do these two bacteria do to each other?
Yeah, so when they co-occur in the lung, our current working model for how co-infections happen in people with CF is that they'll initially be infected with staph, because like I said, younger people tend to have staph in their lungs. And then as they age, people with CF are more likely to encounter Pseudomonas and may get infected with it. So not only can both of these bugs be drug resistant, so that makes it challenging to find a treatment course that's effective for patients with both pathogens. But also people who have both staph and Pseudomonas are observed to have higher rates of pulmonary exacerbation events, which is where they get really sick and need to go to the hospital. And then they also see a more rapid decrease in lung function. So we'll be needing a lung transplant or something similar very sooner if they have both.
And so when you were studying these two bacteria together, did you look at humans, or did you use an animal model? Did you use existing data?
Neither. I really simplified things down, and I wanted to know, outside of the host environment, outside of CF sputum, how is Pseudomonas alone influencing the way that staph evolves and adapts? So there's kind of a couple ways to approach studying pathogens in humans. We can try our best to replicate those environments that they're encountering in the host, in the lab. And there are lots of people working on that. But I was sort of interested from a microbial ecology and evolution standpoint, what is the outcome of a new species being present when a population is evolving? So sort of simplifying things down.
And what did you find?
So I did an evolution experiment actually the summer of my first year. So I was fresh in the lab and didn't really know what I was doing or what I would find. But I was really pleased to see that after just 10 days or so of repeatedly growing staph and Pseudomonas together, I saw that a few of my populations had greatly increased their ability to survive with Pseudomonas. So I was getting much larger staph populations out of the co-cultures where I combined the two together. And so with that finding, we then looked at the whole genome sequences of a few isolates from one of the evolving populations of staph. And we found a surprising result that there was actually only one gene that was mutated in this population that we didn't see in the ancestor or any of those populations that hadn't really changed their survival. And so something about this gene is really important. When I initially looked to see if there were fitness costs associated with turning this gene off, there's nothing that really jumps out. The GLTT mutants, or the aspartate transporter mutants, are just as well able to grow in the lab and infect mice even. And we don't see anything different when we look at them microscopically too. So it's still kind of a mystery as to what exactly is happening here. But I think it's really interesting that we found something that is so, conserved in staph and nature, really strongly selected for in the lab. So I think that tells us that we have a lot more to learn about what staph and Pseudomonas are doing together and what they do to each other.
All right. So there is a gene, but it remains a little bit of a mystery. But what we do know is that these interactions that happen both in dishes and in the lungs of real people are important and could actually cause more disease.
Certainly.
Yeah. Very well. Well, Ashley, it was wonderful talking to you and to learn more about your research.
Thank you, Jaap.
We hope you enjoyed this episode and we look forward to seeing you again. Thank you for watching.